Hello everybody, welcome to Deeksha Online NEET Crash Course Biology. Now after the synopsis, we are going to discuss about some of the important practice questions. Let us first look into the practice questions from the chapter Plant Kingdom. The plant body, first question says, the plant body in the moss is a moss that is Funaria. Now moss is a bryophyte. So if you remember, we had discussed in the synopsis that mosses exhibit a haplodiplontic life cycle. So the main plant body of the moss is predominantly a gametophyte, but it may have a sporophyte attached to it, because please remember, a sporophyte is dependent on the plant gametophyte. Sporophytic generation is dependent. So the answer is, it is predominantly a gametophyte. All of you remember, gametophyte is the haploid generation and the sporophyte is the diploid. Especially since we are talking about the moss, the gametophyte is the leafy generation, and the sporophyte, as you all know, it has a capsule, a seta, and a foot. Next question, the pyrenoids are made up of. We had discussed this also. Pyrenoids are starch granules, actually, storage products which are found mostly in green algae such as Chlamydomonas. So they have a proteinaceous center and a starchy sheath that is surrounding them. So basically, pyrenoids are storage material. Which of the following plant kingdom is called, which of the following plants in plant kingdom are called amphibians of the plant world? A very easy question. So which one do you think is the answer? Yes, it is Bryophyta. So bryophytes are amphibians of the plant kingdom because they live in moist areas and they also depend on water for reproduction. Next question, brown algae is characterized by the presence of which of the following pigments? Now in brown algae, we see a pigment that is referred to as fucoxanthin. So all of you remember, along with fucoxanthin in brown algae, you have chlorophyll A and chlorophyll C, and fucoxanthin is the specialized pigment that is unique to Phaeophyceae or brown algae. Bryophytes can be separated from algae because, so how do you distinguish bryophytes from algae? Now one of the most important difference between bryophytes and algae is bryophytes are the first ones to possess the jacketed sex organs, which are referred to as the archegonia. So the answer is, they possess archegonia. So remember, algae do have antheridium and archegonia, especially if you take algae such as chara. They have structures called globule and nucleus. Globule is the antheridium, nucleus is the archegonium, but they don't have a sterile jacket of cells surrounding them. But bryophytes are the first organisms that possess a sterile jacket of cells around the antheridium as well as archegonium. Now the next question is, heterospory and seed habit are often exhibited by possessing. Now heterospory means the sporophyte, the main plant is a sporophyte. We had discussed, if it is a sporophyte, it has a diploid constitution, chromosomal constitution. It produces spores through meiosis. So it undergoes meiosis and it produces spores. But heterospory means the spores produced are of two types. Some of them are called microspores, some of them are referred to as macrospores. And what is this seed habit? If you remember, all of you, I had told you that in a plant, say for example, this is a plant which is a sporophyte. It is a sporophyte which has a sporophyll, a leaf which bears a sporangium, and it has a structure whose job is to produce a spore. Now what do we call this structure? Let me call this as a sporophyte first. So this is a sporophyte. Okay, now this sporophyte, the job of this sporophyte is to produce a spore, especially the job is to produce a megaspore. So it is a megasporophyte. So this will become a megasporophyll, because we saw that there are two types of spores here, microspores and megaspores, isn't it? So this leaf becomes a megasporophyll, and this structure which produces the megaspore becomes a megasporangium. we had seen that structures which are involved in spore formation are referred to as sporangium. Now, do you remember in the megasporangium, I had told you if this is the megasporangium, now there is a cell which is there whose job is to produce the spore, and this is called a megaspore mother cell, isn't it? So the megaspore mother cell is the one who is going to produce the megaspore. It is diploid, the entire sporophyte is diploid. And then we had seen that it undergoes meiosis to produce 4 haploid megaspores, isn't it? So it, remember, the division is meiosis. So what are these cells which are formed now? These are called the megaspores. So the megaspore mother cell that is sitting inside the megasporangium underwent meiosis to form the megaspore. Usually one megaspore is what survives. So one of the megaspores is surviving, but it does not want to come in, isn't, I mean, it does not want to come out. Usually the megaspore has to come out, but the megaspore is sitting inside the megasporangium, and while it is sitting inside the megasporangium, the megaspore will start developing into a cellular structure which will uh, be referred to as a female gametophyte. So the megaspore becomes a female gametophyte. A gametophyte is a plant which has sex organs, and here it has a female sex organ. It is haploid because it came from the haploid megaspore, but it is inside. Where is it? It is sitting inside the megasporangium. And where is the megasporangium? The megasporangium is attached to the mother plant. I am talking about the same megasporangium here. It is attached to the plant, and inside the megasporangium, the megaspore said, I don't want to come out, I am going to stay inside, and it develops into a multicellular structure inside with a sex organ which is referred to as a female gametophyte. Now, this habit where the megasporangium consists of a developing female gametophyte within is referred to as the precursor for the seed habit. If you go through the synopsis, you will understand what is the true meaning of seed. So I've just given you the simple explanation, and this is seen in a plant that is called Selaginella in Pteridophyta. And which is a characteristic feature of Selaginella? A small leafy structure that is found on the inner angle of the leaves of Selaginella, that is called the ligule. So for example, if this is the leaf of Selaginella, and this is the branch from which the leaf is originating, along the inner margin you will find another leafy outgrowth which is referred to as the ligule, which is a characteristic. The exact function of the ligule is not known. It is mostly a morphological feature, and this is seen exclusively in a plant that is referred to as Selaginella. Okay, so please remember there are two types of sporophytes in Selaginella. That is, one of the sporophyte which bears megasporangium is called a megasporophyte. One of the sporophytes which usually bear. Now here I spoke about the megasporangium. Now sorry, in this there are no two different sporophytes. Now see here, I'm going to modify this diagram here. There may be one more leaf somewhere on the plant which has another tiny structure on it whose job is producing microspores. So we call this particular sporangium whose job is the production of microspores, as we call it as microsporangium. So the same sporophyte has both mega and microsporangium on them. The microsporangium produces the microspores which are the precursors to the male gametophyte, and I told you the story of the megasporangium here, how the megasporangium, it has a megaspore mother cell sitting inside. The megaspore mother cell forms megaspores. The megaspores do not come out inside itself. Inside the megasporangium, they develop into a female gametophyte. A female gametophyte is a plant generation which has the female sex organ. So if you can imagine, a megaspore is giving rise to a female gametophyte, so obviously a microspore will give rise to a male gametophyte which is capable of generating male gametes. Okay, so the answer is ligule. An alga that is very rich in protein is a unicellular non-motile alga which is referred to as chlorella. It is used as a protein supplement, okay? Single cell protein. Largest sperm in the plant world would be in, now largest top-shaped multinucleate sperm are seen in cycas, that is, this is the fruitless but seed-bearing plants which are referred to as the gymnosperms. Okay, so the answer is gymnosperm. Which of the following statement about cycas is incorrect? Now here the incorrect statement is the xylem is composed of xylem vessel. See, xylem vessel is something which is an advanced feature. Okay, xylem vessels or trachea are only found in angiosperms, whereas cycas is a gymnosperm. You mostly find narrower xylem tubes which are referred to as tracheates. So the second option is incorrect with respect to cycas. Next question, dichotomous branching is seen in case of liverworts. Now we had discussed about the thallus of a liverwort, how the thallus of a liverwort, it keeps branching, isn't it? So it branches in twos at every regular interval. You can see it grows further, again it branches into two. So this kind of a branching where you can regularly see branching happening is referred to as, in twos, is referred to as dichotomous branching. This is a characteristic of the prostrate thallus. Notice that this plant body is not differentiated into a root, a stem or a leaf, and it grows horizontally on the surface of the ground, and that's why we say it is a prostrate thallus. Is seen in the case of liverworts. The example for liverworts is Rixia and Markantia. Okay, so the answer is liverworts. The endosperm of gymnosperm, we had learnt about the endosperm of gymnosperm. It is a haploid structure, because all of you remember one thing, in gymnosperms, unlike angiosperm where endosperm is a post-fertilization structure, in gymnosperm, endosperm represents the female gametophyte. What is the female gametophyte? Now, just like what I talked about in Selaginella, here also there is a, in mostly in case of gymnosperms, what happens is, if I take cycas for example, a plant called cycas for example, the main plant is a sporophyte, that is, it is diploid. Now, on one of the leaves of the main plant, there will be, like I told you, if it is a sporophyte which is involved in production of two types of spores generally in cycas. One will be a microsporophyte and one will be a megasporophyte. Usually the female one is the, we should not call a sporophyte as a female sporophyte, because a sporophyte is an asexual generation, but for our convenience the female sporophyte is a megasporophyte. So once again the story goes as, there is a structure on the megasporophyte whose job is the production of megaspore, and this structure is called the megasporangium. Now inside the megasporangium, who is sitting? There is a cell wanting to give rise to megaspore, and we will call this cell as the megaspore mother cell. Is it haploid or diploid? This entire plant is diploid. Okay, now this cell, the megaspore mother cell, will now undergo meiosis. So which plant are we talking about? The main plant is a megasporophyte, which is, for our convenience, we are calling it the female plant. You will see why. So when it undergoes meiosis, it forms a haploid cell. One of this haploid cell survives, and what is this haploid cell called? It is called megaspore. This is the megaspore. It is haploid. And now actually under ideal conditions, the megaspore should come out. It should get dispersed outside. It must get dehisced and dispersed. But in this case, it decides not to come outside. It is sitting inside only, and inside only the megaspore develops into a multicellular structure. It is saying, I am not going to come out, I am going to stay inside, and it develops into a multicellular structure. What is this structure called? Always remember, a megaspore develops into the plant, the sexual generation, which is the female gametophyte. So this female gametophyte that we call in case of gymnosperm as endosperm. Okay, so what is endosperm now? It is a female gametophyte. And where is it sitting? What is this outer structure? This outer structure, we called it as the megasporangium. It is the vessel in which the megaspore was formed, and the megaspore does not come out inside only, it develops into a sexual generation. Which is the sexual generation that megaspore gives rise to? It gives rise to a female gametophyte. In case of cycas, in case of gymnosperms, we call this sexual generation, or the female, not the male, because it's coming from megaspore, we call it as the endosperm. Okay, and this endosperm within the female, that is the megasporangium, may contain 3 to 6 female sex organs. The female sex organs are referred to as the archegonia. So now you know that because it has the female sex organs, and because the female sex organ has egg inside it, we can say that this is now the female sexual generation, that is called the female gametophyte. Okay, so if you say there is a sporophyte which is producing megaspore, the megaspore will always give rise to a female gametophyte. And if there is a microspore, there may be a sporangium on the same plant or on a different plant which produces microspore. Just like megasporangium, there will be a microsporangium. Microsporangium will have microspore mother cell. They will produce by meiosis microspore. A microspore divides to give rise to the sexual generation, that is called a male gametophyte. So don't forget these basics. A megaspore germinates into a female gametophyte. A microspore germinates into a male gametophyte. And don't you think the megaspore is permanently trapped inside the sporangium? In case of gymnosperms, it is saying, I am not going to come out. I am always sitting inside this vessel. Which is this vessel? The megasporangium. Okay, so let me write here, the megaspore is permanently, this is what is the seed habit. It is permanently retained. It is trapped. It is not going out. Normally it should go out and develop into a free female gametophyte. Here it is permanently retained inside. Which is the vessel that produces it? The megasporangium. But unlike the megaspore, the microspore is free. So the male gametophyte is set free. So it is a free structure or a plant generation, whereas the female gametophyte is trapped inside the megasporangium because the megaspore decided not to come out. Okay, in which of the following would you place the plants having vascular tissue but lacking seeds? Which is it? Pteridophytes. Now because there are no seeds, we call pteridophytes as cryptogams. And pteridophytes, as you all know, are the first generation of plants, the first type of plants which develop the ability to live on land, and they are the first ones to, they do depend on water for reproduction. They do live in moist places, but not in comparison as much as bryophytes, and they grow with vascular bundles inside them. They are the first vascular plants. Okay. Plant group with the largest ovule. Again, ovule means, what is an ovule? It is nothing but a megasporangium. I told you, a sporophyte, a plant may be either producing microspores or megaspore. Now, a ovule is a structure that is seen in the megasporangium, in the megasporophyte, and it has the largest tree and the largest gametes. We already saw top shaped multiciliate gametes. It is a feature of gymnosperma. A student observed an algae with chlorophyll A, chlorophyll D. It should be chlorophyll D and phycoerythrin. Which group should it belong to? Obviously, it should belong to Rhodophyta or Rhodophyceae, that is red algae, because these are along with red or R phycoerythrin. You may also find, sometimes they may also give you R phycocyanin. So phycoerythrin plus phycocyanin are the pigments which are unique to red algae, which are Rhodophyta. Sexual reproduction in Spirogyra. Spirogyra belongs to which class in algae? It belongs to which division? Chlorophyceae, isn't it? So now if you talk about the sexual reproduction, we say that it is more advanced. Now it is more advanced because they have physiologically different, they have different sizes of physiologically different sex organs are seen. This is in the case of, the sex organs look very much alike. For example, if you take a Spirogyra filament, this is a cell. Okay, now this could be a cell, this could be a sex organ. It will look exactly like the other sex organ. On what basis are we calling it sex organ? Because now ultimately a tube will be formed between the two, and one of the cells will get rounded off into a male gamete, and it will slide through this tube and it will pass into the other one, which gets rounded off as a larger female gamete, and ultimately both of them will fuse to form a diploid zygote. So what do we call such gametes which are non-motile? We call them as aplanogametes. And we call this mechanism where now what name do we give these sex organs? The male gamete, the male sex organ is nothing but a cell, the protoplast. So we call it as the male gametangium. We use the word for the sex organs here as gametangium. Okay, but it is not jacketed, and this process where a tube is formed between the two sex organs or gametangia, and one of them gets rounded off into a male gamete and slides through the tube and enters into the neighboring cell, which is rounded off as the female gamete, and ultimately both of them fuse together. This phenomenon which I have shown you here, a part of reproduction in Spirogyra, is called lateral conjugation. I hope you know Spirogyra has a filamentous structure like this, where there are cross walls between the filament, and each one is a cell of the filament which have spiral ribbon shaped chloroplast in them. Okay, so it is physiologically differentiated. They look exactly alike, they're morphologically not differentiated, but one of them behaves as the male, one of them behaves as the female. That's why we say that they are physiologically differentiated sex organs. Now moving on to morphology of flowering plants. We are done with plant kingdom. Prop roots are modified into adventitious roots. Now prop roots are actually what? If you see, the prop roots are actually adventitious roots only. But among all of the following, you see prop roots in ficus, that is fig tree. But the prop roots which you see in fig tree are not as prominent as the prop root that you would see in case of a banyan tree. Many of them may not grow up to the ground and they may not form a pillar. Okay? Prop roots are basically roots which are modified for what? For mechanical support. Sharp pointed curved hooks of bignonia. Now in bignonia what happens is it has a compound leaf. Compound leaf means you know about the leaf being dissected into individual leaflets. So this is a compound leaf. It has two leaves at its end, and at its corner it has three curved hook like structure which is like a claw which holds on to a substratum. So this is this kind of a leaf which otherwise would look something like this. So it's as if these three terminal leaflets are modified into these hook-like structures. We call this kind of leaf as pinnately compound leaf. So in this plant which is called bignonia, in bignonia we call the plant as cat's nail because these three terminal leaflets have modified themselves into these hook-like structures which hold on to a support, and this is seen in the case of bignonia. So it is leaflets or the terminal leaflets of a compound leaf blade which is modified into a hook-like structure. I hope you know we had discussed that if you have a leaf which has a dissected margin, but the dissected margin does not reach the midrib, or if you have a leaf with an entire lamina, then we call it as a simple leaf. but here you can see how the leaf is so heavily dissected that ultimately the leaf gets divided into individual segments. And what are we call these individual units of a compound leaf? We call it as a leaflet. Such a leaf where the leaf is dissected into leaflets is referred to as a compound leaf. Alright. In lemon, the petiole is, now we say that in lemon, the leaf is of palmate. We say unifoliately palmate type of compound leaf. So remember, it is unifoliately palmate. And if you see the leaf of a lemon, now the base of the leaf has an expanded part like this, and this expanded part is the petiole of the leaf. So we call this as the winged petiole. So in lemon, we say that is in citrus, the petiole has extensions, wing-like extensions on both sides. So we call it as winged petiole. Identify the plant given below in which you have phyllodes. Phyllodes are nothing but flattened leaf bases. So there is a phyllodes like this, which is flat, and then you have the remaining parts of the compound leaf that you can see in this particular plant. Later, this compound leaf portion will get shed off, and what is left behind is only this flattened green portion, which is actually a flattened stalk of the leaf, which is called the petiole. This is seen in the case of Australian acacia. It's a leaf modification where the petiole itself becomes very broad and it becomes photosynthetic. The actual part of the pinnately compound part of the leaf will basically be lost. Okay. Identify the correct statement. Now the correct statement here is pear is a polycarpic plant. It flowers more than once, and biennials are flower only once during their lifetime, ideally, because biennial means it grows once in 2 years. Now usually what happens is in a biennial, the first year is it shows its vegetative growth, and second year it flowers, and generally the plant dies. So we say both A and C are correct options over here. Simple umbel as a flowering arrangement, it has what kind of arrangement in simple umbel? It is centripetal arrangement, because in simple umbel there is a flowering stalk and all the flowers are borne on the apex of the flowering stalk, and there is an involucre of bracts over here wherever you see the flowers, and it is seen that the oldest flowers are towards the periphery and the youngest flowers are towards the center, meaning growth is happening from the periphery to the center. When growth is happening from the periphery of the umbellate cluster to the center, we say that it is a type of centripetal growth. Okay. In helicoid cyme, the flowers arise on the same side or in all directions. Now actually in helicoid cyme, I think there is some difference in the answer key here. The flowers actually originate on the same side. Now helicoid cyme is an example for a uniparous, we had learnt, or monocaseal cyme. So what happens in uniparous or monocaseal cyme is, if this is the flowering axis, it ends in a flower. You know, cymose inflorescence ends in a flower, and now there is a lateral branch produced on one side. This is another flower. Now this floral or this floret will again bear a lateral branch in the same side. This will again bear a lateral branch on the same side, again on the same side. See, so where is the branching happening? The branching is happening only on the right side of the corresponding floral axis or the corresponding floral branches. This is called as helicoid cyme. Okay, helicoid cyme is seen in Hemilia, begonia, some of the examples in which you can see this kind of a helicoid cyme. Flower arrangement in a biparous cyme is, now biparous cyme means there is a floral axis, the rachis or the peduncle, it ends in what is referred to as the flower. The oldest flower is on the top and then it may produce two lateral branches, one on the right and one on the left. So first you have the oldest flower and then you have younger flowers before them. This is called, because the branching occurs both on the left side and the right side of the main floral axis, we call it as biparous or dicaseal cyme. And what kind of an arrangement is this, where the older flowers are at the apex and the younger flowers are at the base? We call this type of arrangement as basipetal succession. Okay. Now see, they have given some inflorescences here. Now in this inflorescence, the first one looks like racemose inflorescence, isn't it? You can see there is typical acropetal succession over here. But imagine this is not the flower, this is the meristem. So it can grow continuously upwards. It is capable of growing throughout, and older flowers are towards the base and younger flowers are towards the apex, and all the flowers have stalk. They have a stalk or the pedicel. So we can call this as a simple raceme that is seen in Crotalaria, Delphinium, Snapdragon, isn't it? So all these are some of the examples. Most common example would be Crotalaria, in which you will find this kind of a simple raceme. Now here, this is also a racemose inflorescence. It is not ending in a bud, but it grows continuously. But notice that all the flowers are without a stalk. See, and there is bract at their, they emerge from the axil of a bract. So we call this type of inflorescence as a spike inflorescence. Now in which one did we see spike inflorescence? In Acheiranthus, in Adathoda, in Callistemon, we saw spike inflorescence. Now here, this is a uniparous cyme because this is the axis ending in a flower. Only one branch is produced on the right side. Either sides, it may be right or left, but here only one branch is produced. And here, this is a polycaseal or a multiparous cyme. It ends with a flower and each one of the branches are biparous cymes in turn. See, it is a biparous cyme which has branches of biparous cyme as well. So this is seen in the case of Calotropis is the best example. Nereis, Calotropis, Viburnum, all these are examples for polycaseal. And unicaseal, monocaseal or uniparous, we saw it, if it is branching on the same side, it is helicoid. If it branches on alternate sides, then it is called scorpoid. So the correct answer to this question would be, that is racemose inflorescence. The second one is spike. The third one is uniparous or monocaseal, and the fourth one is a dicaseal cyme, which is a compound dicaseal cyme, or a polycaseal it should be actually. So this should be polycaseal because each branch is further exhibiting a dicaseal branch in it. So the correct answer would be option B. Tuberous roots are thick and fleshy. Now they are talking about not the tuberous roots of sweet potato. The tuberous roots of sweet potato are adventitious root, but sometimes a tap root may also become irregular in shape. So tuberous roots which are tap roots which become irregular in shape are found in the 4 o'clock plant, that is Mirabilis jalapa. Taxonomical terms for the following aestivation. Now this one would be where you notice how aestivation means the mode of arrangement of calyx or sepals or petals in a bud condition. So this is called valvate aestivation, this is twisted aestivation, this is imbricate aestivation. Okay, and here you can see two types of imbricate. One is, and notice here one petal is completely outside, one petal is completely inside. So let us see what the options are. It is valvate, twisted, imbricate and quincuntial. Imbricate means there is no regular pattern of arrangement. Quincuntial means one of the petal becomes completely outside and one of the petal becomes completely inside. Identify the fixation of the filament. Now here you have adnate. So if it is attached like this throughout the, see, there is throughout, the filament extends throughout the two lobes of the anther. We call such an anther as a dithecous or a bithecous anther because it has two lobes on its side. If it is attached only to the base, then it is referred to as the basifixed. If it is attached to the back, it is dorsifixed. If it is attached at 2 points, and actually in case of versatile, you can see that the filament is attached to the basal side, and the two lobes of the anther are very free. They keep moving in the air, they sway in the air like in the grasses, so they are relatively very free. That's why it's called versatile. Okay, so the option A is the correct answer. Infundibulate corolla means you have a flower in which the petal is in the form of a funnel like structure. So we say that funnel shaped corolla where all the petals are fused. Obviously, what do you call this condition where the petals are fused? It's called gamopetalous, and all of them are fused to form a tube like corolla which is in the form of a funnel shape, and that is what is called infundibulate corolla. Okay, if it is bell like, then it is called, for example, you have fused corolla like this which has a bell like structure. If it is like this, then we call it as campanulate type of corolla, but here the answer is infundibulate or funnel shaped. Didynamous condition. Didynamous condition means there will be four stamens. Okay, the two of the stamens will be taller and two of the stamens will be shorter. This is called didynamous stamen, which is seen in ocimum, in salvia, etc. Okay. Mericarps and cocci occur in dry fruits. Actually, the name of the fruit in which you can see such units called mericarps or dry cocci are called schizocarpic or schizocarpic fruits. So what happens here is, imagine if the fruit develops from a trilocular syncarpous ovary which has three seeds in each of its locule. Then during its dehiscence, what happens once the fruit is mature is it splits along the septa. See, these partitions are referred to as the septum. So the fruit is known to split along the septum, and they get separated from each other into single seeded units, and these single seeded units are referred to as cocci or mericarps. And then the cocci breaks open and the seed comes out of it. Okay, this kind of a fruit where the fruit breaks along the septa. See, the fruit has splitting, that's why it's called schizocarpic. It splits along the septa, and each unit of this fruit, of a dry schizocarpic fruit, is called a cocci or a mericarp, and this is seen in the case of regma. Example for regma is castor and jatropha. Castor is Ricinus communis, so the answer is regma. Okay, now moving on to the next chapter, that is biomolecules. Phosphodiester bond is always formed between the third carbon of one sugar and the fifth carbon of the next sugar in the nucleotide, in the polynucleotide chain. So please remember, it is always between, say for example, this is the sugar, the pentose sugar, this is the fifth carbon. The fifth carbon is connected with a phosphate group to, let me show the next sugar very tiny, there's no space here, to the third carbon of the next sugar. So this is the third carbon and this is the fifth carbon. Okay, so we say that this kind of an arrangement is what we see in the case of phosphodiester bond. Glucose concentration in blood in a normal healthy individual is 4.5 to 5.0 millimoles. Study the following ionizable state of amino acids. So here you have a state which I want you to focus. Notice that this amino acid has a zero net charge on it because it has positive charge on its amine group and a negative charge on its carboxylic group, and this is referred to as a Zwitter ion. So it is the second option. In primary structure of protein, the left end is represented, the right end is represented by the last amino acid. So it is the third option. Now here, all of you remember, I had shown you the beaded chain here. Each bead is nothing but the amino acid. So imagine there are 1, 2, 3, 4, 5, 6 amino acids. This is the first amino acid and this is the sixth amino acid. The first amino acid is on the left end, the last amino acid is on the right end. So the right end is represented by the last amino acid in the polypeptide chain. The correct pair of biomolecules, one of which is an aromatic amino acid, and whereas the latter is a toxin, rixin is a toxin. So phenylalanine is an aromatic. Histidine, tryptophan, tyrosine, phenylalanine, all of them are aromatic. So the correct answer is C, because rixin is a toxin that is obtained from castor plant, that is Ricinus communis. Adult hemoglobin consists of, it has 4 subunits of 2 types. Out of the 4 proteins that it has, 2 of them are referred to as alpha and 2 of them are referred to as beta. Which of the following are components of phospholipid? We had learnt in the morning in the synopsis class that a phospholipid consists of fatty acid along with glycerol, the trihydric alcohol, but it has a phosphate group. Of course, along with the phosphate group, it has something called as an additional group also. So remember, it has a phosphate group, an additional group, fatty acid and glycerol. So the second option is the correct answer. The nucleotide sequence here, obviously if they've given one strand, you should all remember that the second strand they're asking you the sequence of. The second strand is anti-parallel. It runs in 3' to 5' direction, and then as per the base rule you write the bases. Adenine pairs with thymine, thymine with adenine, guanine with cytosine, cytosine with guanine, cytosine with guanine, adenine with thymine, and so on. So if you do this, you will get the correct answer, that is the last option, which is the correct complementary strand. The R group in serine and alanine are CH2OH and CH3. Please remember, if you take an amino acid, it has an R group, an H group, it has an NH2 or an amine group and an acid group. R group varies from one amino acid to another, and that's why the R group is called a variable group. So for serine, the variable group is hydroxymethyl. For alanine, it is methyl. If you remember, for glycine, it is just a hydrogen. So glycine is an amino acid which has 2 hydrogen atoms attached to it, because even the R group is a hydrogen and amino acid already has a hydrogen attached to carbon. So the simplest amino acid, however, will be glycine. Storehouse of energy in animal tissues. Which is animal starch among the following? Glycogen is the animal starch. The most abundant elements in the protoplasm are carbon, hydrogen, and oxygen. Very basic question. Fructose is a sugar which has a ketone group, and it is a sugar which contains 6 carbon in it with a ketone group in it, and that's why we say fructose is a ketohexose. Which amino acid is required for the synthesis of hemoglobin? Now there are many amino acids, but in the order of requirement, it is said that histidine is an amino acid that is most important in the synthesis of hemoglobin. Cellulose eaten by grazing animals is passed out undigested, digested by gut bacteria, digested by digestive enzymes, and intestinal bacteria. You all know that in grazing animals there are bacteria, as well as they may produce what? Enzymes, digestive enzymes. But you know that in the stomach, called rumen, reticulum, omasum and abomasum, mostly fermentation takes place. So it is intestinal bacteria which basically help in digestion of cellulose. Okay. Which of these is not a polysaccharide? Glycogen, amylose, cellulose, chitin, glycogen, amylopectin, cellulose. Now see the second option, we have a monosaccharide here. That means this group does not include only polysaccharide. So the answer is second option. Now let us move on to the last chapter in this week's class. We have cell cycle and cell division. Let us quickly run through the stages. Okay, so now obviously they've given you all the stages. Let us mark them one by one. So first one is prophase. See how it is early prophase where the nucleus has still not disappeared, but here you can see how in the late prophase the nucleus has started disappearing, isn't it? And then after that you can see there is metaphase over here. The chromosomes have come and aligned at the equator, correct? And then it is the fourth phase. They're beginning to get, suddenly the cell has been tilted to the right, so note the change. They're starting to get pulled away during anaphase, and they have become pulled away quite far away from each other. So obviously it is the correct sequence which I have marked here. It would be the second option. Okay, now division of nucleus is referred to as karyokinesis. Division of cytoplasm, it is referred to as cytokinesis. Replication of DNA occurs during S phase, which is during interphase. If karyokinesis happens but cytokinesis does not happen, we had seen that there is formation of a multinucleate mass of protoplasm, which is referred to as syncytium in animals, cenocyte in plants. So this is the correct option here. So it is the option B among the given options. Please refer to the match the following once again. It's not in the same slide. A cell is a continuous process in the cell. I think there is some discrepancy here. Probably cell division they're asking is a continuous process in the case of cell cycle. It occurs in G phase S1. It should be very specific here. G1, S2 and G1 S phase or synthetic phase and G2 phase or M phase. Let us see what the answer is. It says both A and R are true, and R is the correct explanation for A. So probably from the answer I am able to tell you that cell cycle, or we can say cell growth, maybe they mean, isn't it? Because throughout the cell cycle the cell keeps growing and their cell is metabolically active. So now it makes sense, cell growth is a continuous process in cell cycle. And why do we say it is a continuous process? Because it has all these events which are joined end to end, that is gap 1, synthetic, gap 2, and the actual phase when the cell divides, which is referred to as the M phase. Okay, synthesis, that is DNA replication, does not occur throughout the cycle, and DNA replication is a discontinuous process. The first one is true, it occurs only during S phase. It doesn't happen throughout the cycle. And no, DNA replication or synthesis is not a discontinuous, it doesn't happen in breaks. It is a continuous process. So option A is true, but R, that is assertion, is true, but reason is false. If the endosperm cell of a plant has 36 chromosomes, okay, first we should say what is the number of chromosomes in the each pole during anaphase. Okay, now let us say, now please remember they have said anaphase 1, so they are talking about meiosis 1. Now first of all, endosperm is 3 sets. So if 3 sets is equal to 36, one set or diploid set, let us not take one set here, let us take diploid set, is equal to 24. Correct? Now imagine that this diploid cell is undergoing meiosis. So to give you the example, let me take a small cell with only 4 chromosomes in it. 2 longer chromosomes and 2 smaller chromosomes. So 2n is equal to 4. Now during S phase the chromatids get duplicated. So here you are seeing only 1 chromatid per chromosome, but now you will see 2 chromatids per chromosome. That means S phase DNA synthesis has happened here. See, so this is S phase, 2n is equal to 4. And then now see what happens here. S phase 2n is equal to 4. Now during anaphase 1, all of you remember, during metaphase 1, the pairs will come and align at the equator like this. The taller ones and the shorter ones. Both of them are in the equator, and anaphase, what will happen? They get pulled away from each other. The pairs get separated. So let me draw anaphase here for the same cell. Now let us count how many chromosomes are there in each pole during anaphase. We started with 2n is equal to 4 diploid cell, and we have 2 chromosomes, half of 4. So if you start with 2n is equal to 24, in anaphase 1 you will have 24 divided by 2, 12 in the upper pole and 12 in the lower pole. That's why the correct answer is 12. So they have given this endosperm thing only to confuse you. You should remember that the endosperm has 3 sets of chromosome, and in a diploid cell you have 24 chromosomes. That is, if 3 sets are 36, 1 set is 12 and 2 sets or diploid is 24. It is usually a diploid cell that undergoes meiosis. So I took an example of a small cell with 4 chromosomes, and we saw in anaphase 1 there are 2 each per pole. So if we start off with 24, we will have 12 per pole. Okay. What is untrue about telophase? Now in telophase phase, disappearance of nuclear membrane is not true, because actually in telophase, nuclear membrane reappears. Chromosome undergoes decondensation, that is, it starts becoming thread-like, it starts losing its chromosomal nature, its condensed nature, and there is disappearance of the nuclear membrane. Okay? Sorry, reappearance of nuclear membrane, which is why disappearance of nuclear membrane is false. In which order cytokinesis occurs in plants? Now cytokinesis occurs in centrifugal order. Centrifugal means away from the center. Okay. Genetic information is transferred from zygote to all the body cells by mitosis. So please remember that mitosis is what is involved in the case of transference of genetic material, because the zygote divides by cell division and it undergoes mitosis. Mitosis helps the plant in. What does the mitosis do? It helps in conserving the genetic integrity of the whole. It maintains the chromosome number. That's why we say conserves the genetic integrity. A cell divides every minute. It will fill 500. It will fill a 100 ml beaker in 1 hour. Now, what do you mean by a cell dividing every minute? You start with one cell. In 1 minute, it became 2 cells. Or if you have 2 cells, when was it 1 cell? 1 minute ago. 1 minute ago it was 1 cell. Similarly, if you have 100 ml, it fills 100 ml of the beaker. When will it fill half of 100 ml? That is 50 minutes. 50 ml. At 60 minutes, it has filled the full beaker. So 1 minute ago, it fills 50 ml. So 60 minus 1 minute is 59 minutes. So remember, two ways of looking at it. Either you can say in 1 minute one cell divides into 2, or 2 cell was 1 minute 1 minute ago. Sorry, 1 cell 1 minute ago. Similarly, 100 ml of the beaker was half of it. That is 50 ml. How many minutes ago? 1 minute ago. If it is 100 ml at 60 minutes, 60 minus 1 minute ago will be 59 minutes. Okay? A root apical meristem divides 8 generations. Okay, and they're asking 25% of the cell stops dividing. Then how many cells are there in the root tip? So let us start with one cell. One cell becomes 2, 2 becomes 4, 4 becomes 8, 8 becomes 16. Mitosis, remember, 32, 32 becomes 64, 64 becomes 128, 128 becomes 256. How many generations? This is one generation, second generation, third generation, fourth generation, fifth generation, sixth generation, seventh generation, eighth generation. So we have with us now 256 cells. 25% stops dividing. So 25% of 256 is equal to 64. So 64 cells have stopped dividing. So let me subtract 64 from 256. I am left with 192. 192 cells are still dividing. They are still very much active in dividing. And they divide for how many generations? 2 generations. That means 192 in one generation becomes double 384. 384 becomes double 768 in the second generation. So now I have 768 new cells plus 64 which had stopped dividing. So 768 plus 64, the answer will be 832. Okay. In angiosperms, when does meiosis happen? Now meiosis happens during the formation of the male and the female gamete. Okay, so the answer says pollen grain or microspore formation. I'm sorry. Yes, male and female gametes are formed from the pollen grain by male gamete specifically is formed from the pollen grain by mitosis. You have a microspore mother cell which is diploid, which undergoes meiosis, and this produces haploid cells which are referred to as microspores. Each microspore will develop into a two-celled structure that is called pollen, generative cell and vegetative cell. A bivalent means, if you remember, there are one pair of chromosomes with two chromatids each, which form a pair. This is called a tetrad or a bivalent, isn't it? So in bivalent, we have how many pairs of chromatids here? So this is one pair of non-sister chromatids. They are saying one pair means two non-sister chromatids. So here two pairs of non-sister chromatids. So these two are non-sister chromatids, these two are sisters, but these two are non-sisters, because if you consider one chromatid belonging to this chromosome and one chromatid belonging to this, they are non-sisters. Similarly, this chromatid and this chromatid are non-sisters. So the answer is two pairs of non-sister chromatids. Okay. Such a pair is called a homologous pair or a tetrad or a bivalent. Synapsis happens during zygotine, where chromosomes come together and start forming a pair. Synapsis is the pairing of homologous chromosomes. That's what gives rise to a bivalent or tetrad. So this completes the practice questions. Please solve many more questions from your NEET manual for the same purpose. It will help you understand the recall whatever basics you had learnt last year. So until we meet next time, thank you everybody.